Now I want to remind you that there are many worksheets available for extra practice. If you go to Canvas Files on Unit 2 and go down to Worksheets, there is two PDFs. Oh, that's the other one. Yeah. And this particular PDF has got about 5 worksheets embedded in it. And they're naming and reactions and just more of the same. Okay. And notice it's all line drawings, none of that C and H stuff. So this is some of the stuff we're going to be getting into today, actually. Okay, so let's. Oops. Okay. All of you know what a carbohydrate is. You've heard of it. It's in the news. Cut down your carb, you'll lose weight. Which is partially true, by the way, not totally true. Some carbs you're required to have, period. Those are the complex ones. But you know, if you look at a nutrition guidance, there's only two major food groups that you need for your body. You know what they are? Proteins and fats. I did not mention carbs because there's a process called gluconeogenesis in your body. If your body needs carbs, it makes them. So there's a very major push right now in the last couple of years in diabetic research on turning around, particularly type 2 diabetes, even in some cases type 1 diabetes, where you totally eliminate carbs from your diet. All you eat is protein and fats. One, you lose weight, and secondly, your body makes the carbs, and it doesn't make excess carbs, makes just enough it needs. Hence, your pancreas functions a lot more efficiently because it doesn't have to make as much. And I stuffed myself last night with chocolate. Anyway, it's a really interesting thing. In fact, that might be a nice project also, if you'd like to study that more. Okay. So everyone knows that we get energy from carbohydrates and fats. And if you're starving, from proteins also. When our body needs energy and we don't have any carbs available, it will take and convert other biological molecules into glucose. That's the gluconeogenesis process I talked about. And then when it makes the glucose, it just goes through glycolysis, Krebs cycle, electron transport, and produces adenosine triphosphate energy molecules. Okay. Frequently, carbohydrates, particularly in workout journals, etc., they'll refer to carbohydrates as CHO, because carbohydrates, 99.9% of them only have carbon, hydrogen, oxygen in them. So they will refer to them as that. So if you see that when you're reading something on exercising or nutrition, that's what they're referring to. There's two ways to illustrate sugars. This is a simple sugar. This is ribose. You've all heard of ribose in RNA, DNA, deoxyribose. For example, this is not deoxyribose. This is plain old ribose. And Deoxy means it eliminates a hydroxyl group and converts it into hydrogen, hence the deoxy. And there's two ways to visualize this. Now, this is with all the C's and H's and blah, blah, blah. Most of the time, we don't look at them like that. We look at them in their cyclic version. And there are no C's and H's, which is basically a line drawing. Now, there are four classes of saccharides we're going to be talking about in carbs. That's the simple ones, the monosaccharides. And you'll have to learn three of those. Disaccharides, you'll be learning three of those. Oligosaccharides, you'll be learning two of those. And polysaccharides, you'll be learning three of those. These are the ones here that frequently are not studied in this class. When I taught this class up in Washington, they didn't talk about oligosaccharides at all. They're extremely important. They're kind of a longer chain saccharide. For example, who's heard of probiotic? Anyone? Okay. And what's that? Probiotic. You've just heard of it, right? Okay. Probiotic talks about the microbiome in your gut. Microbiome is becoming more important every single day. There are studies to show that, for example, the bacteria in your stomach or in your gut is responsible for things like depression, obesity. It goes on and on and on. It's amazing. And that group is referred to as either microbiome or probiotics. Now, frequently in our country, we eat a very limited diet. And because of that, our gut bacteria are very narrow in terms of diversity. So frequently, people will take a probiotic, which is basically a tablet full of bacteria, good bacteria. There is good bacteria and bad bacteria. Now, we have to feed our bacteria, and we use something called prebiotic. And that's food for the bacteria in our gut. If you don't feed them, guess what? They eat anyway. And guess what they eat? They eat the mucous membrane on the inside of your intestine. And you end up with something called leaky gut syndrome. That's holes in your intestine. So if you have something like bad stuff going on in your intestine, like an infection, and you have leaky gut syndrome, that infection can get into your body and you'll end up with sepsis. So it's very important to feed your bacteria. You can even have a test done on what bacteria you have in your gut. I had it done about 2 years ago, and my gut bacteria is fine shape. I eat a lot of vegetables. Those are prebiotics. Normally, complex carbs are prebiotics. Now, to go back to oligosaccharides. Babies in baby formula and baby's breast milk have a special oligosaccharide in there that's a prebiotic for the baby's microbiome. And if you're feeding that baby a formula, you better make sure it's got that oligosaccharide in it. And the baby doesn't digest it. The baby's bacteria digest it. So that class of carbs is really important and frequently overlooked. So I always cover it to make sure. And as health, most of you going into allied health, that's a really budding area to get into. Okay, I've talked about this before. We have geometric isomers. And a complex form of a geometric isomer is called an enantiomer. And that's this right and left hand bit. Okay, again, we've got one palm on both sides. We've got 5 fingers attached to that one palm and basically the same place, but the molecules are not the same. They're mirror images of one another. That's called an enantiomer. And from a biological perspective, it's very important because our body's enzymes will only be specific for certain enantiomers, and they're referred to as D and L. I mentioned a while back the thalidomide problem back in the 60s. That was a D and L problem. MSG, another problem. Some people are very sensitive to MSG. Our bodies make the L form of glutamic acid, which is the glutamate part. We actually make that in our body. So when we take the D version of MSG, that's the unnatural version. And some people are very sensitive to that. And when you buy MSG, you get everything, D and L both. So this distinction here is really important. I remember helping a student study for her MCAT test, which is the medical school entrance exam. And this was a question that was on there. Chiral carbon. Okay, a chiral carbon is a carbon that has 4 unique groups attached to it. Doesn't matter how big they are. They have to be unique though. And it has to be 4, no exceptions. So if we go down this list here, let's look at this simple one first. Okay, so let's look at this first one over here. Let's call that. Okay, so if you look at group number 1, now I'm talking about is that a chiral carbon? Okay, so we have to look at the groups attached to that carbon to determine if it's chiral or not. Okay, so we have this group here. It's an aldehyde. We have a planar hydrogen. We have a hydroxyl group, and we have a methoxy group down here. So is that C in the middle chiral? 2 questions. Are 4 groups attached? Yes. Are they all unique? Yes. So if you answer yes and yes, it's chiral. So therefore, a D and an L form exists. If it's not chiral, no D and L exists. We look at a more complex carbohydrate. This is a hexose, 6-carbon sugar. If we look at carbon number 2, now first of all, this is an aldehyde group. How many groups are attached to that carbon at the very top? Are there 4 attached? One, two, 3. Only 3. So the top carbon is not chiral because it doesn't have 4 groups attached. Okay, what about the next one down? And we've got 1, 2, 3, 4. So it's got 4 groups attached. And are they unique? They are. They're all different. So that means carbon number 2 is chiral. That means that molecule can exist in a D and L form. Okay, let's look at carbon number 3. Okay, we've got 1, 2, 3, 4 groups. That's good. And they're all unique. So carbon number 3 is chiral. Likewise, carbon number 4 down here, we have 1, 2, 3, 4. Again, and they're all unique. So that's chiral, and carbon number 5. Now, carbon number 6 at the bottom and the one at the bottom has got 4 groups, but they're not unique. There's 2 hydrogens there. Hence, carbon number 6 is not chiral. So carbon number 1 is not, because it doesn't have enough groups, only has 3. Carbon number 6 is not, because the 4 groups attached are not unique. So this particular molecule has 4 chiral carbons. Don't worry about this. Just know that you're going to have multiple chiral carbons and how to calculate them or figure that out. Now, normally we use line drawings, not stick figures like this. So this would be what that molecule would look like. Now, there's a standard that we use whenever we draw small carbohydrates, that we put the aldehyde group at the top and the hydroxyl carbon at the bottom. For it to be a D, the OH has to go to the right. Hence the D there. That's the carbon furthest from the top aldehyde. So since this goes to the left, it's an L form. That goes to the right, it's a D form. Now, one of the things that this slide didn't talk about is to be a sugar, you've got to have some kind of a aldehyde or ketone in that sugar. If it only has alcohols in it, hydroxyl groups, then it's called a polyol. And frequently they use those as sugar substitutes. But to be a carbohydrate, you've got to have an aldehyde or ketone in there somewhere. So those are both aldehydes now. Another characteristic of carbohydrates is they will rotate light. So if you shine a polarized light through a solution of carbohydrates, the light will twist. The degree by which it twists tells you how concentrated the carbs are. Now, when I was an undergraduate, Davis, part of my research was on carbohydrates. And the professor I worked under, his specialty was trying to make sucrose in the lab. It was very hard to do. And he was very old and he was legally blind also. So one of the reasons he hired me was for a set of eyes. He told me later. Kind of interesting. And then once he found out that I could spell test tube, he started giving me projects to do. Okay, so enantiomers will rotate light. So when it rotates it to the right, it's called a dextrorotatory. versus a levorotatory. And those begin and end with, begin with a D and an L, but they're not the same as a big D, big L. Okay. Unfortunately, the names are really sound familiar. Okay, now, D and L enantiomers are found a lot in nature. A lot. And since most reactions in nature are catalyzed by enzymes, and enzymes have very specific active sites that can distinguish between a D and an L, the biological systems tend to make only one or the other. Now we're going to get into enzyme structures a fair amount in a later unit, but that's the big deal with D and L. You cannot throw something in your body that is not designed to. That's the whole problem with genetically modified plants and animals. You're sticking stuff in your body that your body's not used to seeing. So sometimes it can react in a good or bad way. Corn. It's very difficult to find corn that's not been genetically modified. It's called GMO, genetically modified organism. And the problem is they grow so much corn in the Midwest that the pollen just kind of blows around. They have a lot of wind in the Midwest, in Iowa and Kansas and stuff. It's really hard to find isolated corn that has not been genetically modified. Now, we've been genetically modifying plants for thousands of years. It's called selective breeding. Not by chemicals, selective breeding. So we selectively breed different kinds of corn to produce characteristics. For example, I used to own a walnut ranch in Winters, 120 acres with 3,500 trees, and we grew the older version of walnuts. The newer version got better yield than our version. And it had other issues, though. It's more susceptible to disease. And so you ended up with the same yield, except there was less trouble with the older versions. But those have been selectively bred through the university for probably 80 or 90 years. So man and woman have been doing selective breeding forever. Just like your dog doesn't look like a wolf anymore because of selective breeding. So there's two kinds of genetic modification: selective breeding and enzymatic modifications. There's a lot of stuff going on right now in our world and on the genetic engineering front. Some of it's really scary and some of it's really cool. Associated also with AI. There's a special on Netflix put out by Smithsonian on artificial intelligence. The good and the bad. The bad is scary, really scary. And the good is cool. I think the bad outweighs the good, though, at least as far as it's serious. But if you have time over break, you ought to watch it. It's very interesting. Let me give you one little snippet out of it. They produced this artificial intelligence program to artificially synthesize different molecules. And in the programming, I used to teach programming, and there's something called a Boolean logic statement. It's either true or false. And it usually gets you to a different path, depending on whether the logic is either true or false. Well, one of the statements in this long AI program was beneficial, yes or no. If that statement was true, the program continued on. If it was no, the program stopped and didn't produce these particular molecules. In other words, they're harmful. Well, what they did as a lark is they changed that to, if it's false, go on. And it produced 25,000 new bioterror molecules. 25,000, like that. And, organic chemists can synthesize them. So, and then the government found out about this laboratory, and I don't know if the CIA or FBI went in and confiscated all this stuff. And they had to go before Congress to talk about what they've done. And they just said it was a lark. It took 30 seconds to go in, find the code, 2 or 3 million lines of code, find the code, change that one Boolean statement. That's all it took. That code could fit on this flash drive. This is not a terabyte flash drive. This is an old one. This, I think, is 8 gigs. So it's really scary stuff. Okay. So bottom point on this slide is that our body is horrendously selective for either the D or L form. Okay, so let's talk about naming. One way to name a simple carb is by how many carbons it's got, and then put OSE at the end. So if it's got 3 carbons, it's a triose, pentose, hexose, heptose, whatever. All that does, though, is it tells you how many carbons are in it. It doesn't tell you whether it's D or L. It doesn't tell you whether it's a aldehyde-based carb or a ketone-based carb. It just says how many carbons are on. So that's one level of naming. How many carbons does it get? Another consideration is that an aldehyde-based carb or is it a ketone-based carb? So when that carb is in its non-cyclic form opened up, it'll expose an aldehyde or a ketone. For example, glucose is an aldohexose. Fructose is a ketohexose. As fructose opens up to a ketone, not an aldehyde. Hence, its chemistry is going to be different. That's the issue with fructose in your diet. Fructose is metabolized differently than glucose. And if you want to look it up, it's at the 3rd stage of glycolysis. Okay, so depending on how many carbon atoms make up the sugar, you'll get different variations on the direction of the hydroxyl groups. So, for example, hexose, 6 carbons, 4 chiral. Now remember, each chiral can be D or L. So that means there are 16 versions of hexoses. 16 versions. And it all has to do with which way the hydroxyl groups are going. Now, you're going to learn some of these. You're not going to learn 16 of them, though. So if you've got 3 carbon atoms, you do not have a chiral carbon there. So hence there's only one form. If you throw in another carbon, you can have chiral carbon in there. In that case, you're having a D and an L form of that. That's where all the different configurations come into play and make things really complex carbohydrate-wise. So here we have the hexoses. And you're going to need to know this one, And these are the aldo hexoses. And you're going to learn a keto hexose, and that's going to be fructose. So those are the three monosaccharides you're going to need to know. You're going to be able to draw particularly glucose like that after a while, because you're going to draw it so much. And I'm going to show you some tricks as to how to remember. What's the difference between galactose and glucose? It's on carbon number 4's hydroxyl group goes up rather than down. So once you know glucose, you sort of automatically know galactose. Fructose is weird because it's a keto hexose. Small sugars are sweet. Big sugars are not sweet. Who cooks in here? Raise your hand quickly. Okay. Who's heard of Caro syrup? You go by the store and look at it, it's corn syrup. So tell me whether that's sweet or not sweet. What's the big trouble when you drink soda? Regular soda. Hmm? Yeah. So what's wrong with drinking regular soda? When you go down to Costco and buy Coke from Mexico, what do they raise the flag saying this has no what in it? High fructose corn syrup. High fructose, but I just said Cairo corn syrup. So I was in the aisle one day, this was about 10 years ago, and I cook a lot, as you all know. And I went by the aisle, and there's this big deal on Cairo corn syrup. They make pancake syrup, and I started going. So I picked up a bottle, and there are no small sugars in there. It's all big, long polysaccharides. High fructose corn syrup, it takes each part of those long chains, they're glucose molecules. And that's where the high fructose comes in. Second step is look at the difference between, well, invert sugar, not invert sugar, sucrose, table sugar is what, 1, the relative sweetness. Fructose is almost twice as sweet. And here's glucose. Glucose isn't that sweet at all, it's only 0.74 relative degrees of sweetness. So the scientist was invented in Japan. So the scientist said, well, not only did we take apart that big, long starch molecule in corn syrup made into glucose, now it's sweet. We can make it sweeter if we convert the glucose to fructose. And they do it enzymatically. So now that high fructose corn syrup is much, much sweeter than plain old sucrose. And we grow a ton of corn in this country. So there's a lot of spare cornstarch around. Invert sugar is what's in honey. Honey, sucrose is made up of glucose and fructose, but they're tied together into one molecule. Invert sugar is fructose and glucose are present. They're just not tied together. They're separate. And they call it invert sugar. Okay, so this is what you normally see. This is what you normally see when you think of a simple sugar, is the cyclic form. And it's called a Hawthorne structure. Okay, it's a line drawing, except for that top CH2. And normally when I do it, I don't put the C in. So the way I would draw glucose, for example, now glucose, this is the way I remember it. The OHs alternate. And this one here, let's draw this a little bit here. That's the anomeric carbon. And the hydroxyl group attached to it can either be up or down. Only on the anomeric carbon, and that's carbon number 1. So this is a hexose, so it has 6 carbons. We start there and go so the important carbons are carbon 6, carbon 4, carbon 1. Those are the 3 spots where major reactions happen in hexoses. There's a lot of exceptions, but probably 95% of the reactions involve one or some of those 3 carbons. Okay, now, we're going to be using D forms of the monosaccharides. So I ask you to name a structure. You don't have to say whether it's D or L, because all of them are going to be D. So you don't have to put the D in there. You do have to put whether it's a beta or an alpha form of that anomeric carbon's hydroxyl group. Okay, whether it's above or below. So and interestingly, sometimes your body will convert between alpha and beta. Now, does it enzymatically? Okay, now, this is a very complex slide. And, oops. Notice the beta, oops. So that means the hydroxyl group here is going up. Down here, the hydroxyl group is going down. Now, this cyclization, what that means is it opens up and exposes the aldehyde. Now, aldehydes are not particularly stable, so they want to react. What happens is it closes back up. And sometimes it's in the beta form, sometimes in the alpha form. So glucose is constantly opening and closing. The cyclic one is more stable, so it spends more time in the cyclic one rather than in the open one. When it's open, it's an aldehyde. When it's closed, it's something else. So let's explore what that is. Okay, first thing we're going to do is going to draw a closed glucose. We're going to go down, up, down, and let's make it an alpha glucose. So that means the OH is going to go down here. Now, remember I said that was the anomeric carbon right there? And remember the last lecture we had on aldehydes and ketones, we talked about acetals, ketals, hemiacetals, hemiketals. First step was look for the OCO. Do we have an OCO present there? We have OCO. So the anomeric carbon, carbon number 1 is anomeric. Okay. So OCO is present, is a hydroxyl group attached to the anomeric carbon. Yes. Is there a hydrogen present on that anomeric carbon? Yes. Because remember, carbon has 4 bonds. On that line drawing, we've only accounted for 3. 1, 2, 3. So that means the other bond is a hydrogen. So number 2, yes means it's a hemi something. Number 3, if it's a yes, means it's an acetal. So hemiacetal. So glucose, galactose, mannose, all these sugars have a hemiacetal present. So that means that hemiacetal can open up to the original aldehyde and the original alcohol from which it was created. What happened? Okay, so this is oh, that's what went out. I thought you were pointing to the lights this time. Okay, so it's on the projector side. Okay, let's turn it off. Well, there's a whole bunch of blinking red lights now. There's a saying, live by the sword, die by the sword. All right, well, guess we're gonna do it this way. Okay, so let's start with glucose again. Okay, so we have alpha glucose. Alpha because the hydroxyl group is going down on the anomeric carbon. Okay, so because it's a hemiacetal, OCO, hydroxyl group, and a hydrogen there, it's a hemiacetal. Okay, so hemiacetals can go back to their native aldehydes and alcohol. What else can go wrong today? Okay, so now we have the original aldehyde from which it was created and the original alcohol. These come together to form a hemiacetal. And this is the anomeric carbon right here. And notice it's a two-way street. So this can open, can close, open, can close. Now, we have an aldehyde present. Hence, it can go through all the aldehyde reactions, like the Tollens reagent, Benedict's test. All those work in this guy here. Because this aldehyde is very easy to oxidize. Aldehydes, we oxidize them, it goes to the carboxylic acid. All right, well, I'm still recording.